art of mushroom consumption by people from all walks of life crosses all cultural boundaries and is as old as civilization itself. Mushrooms are edible, fleshy macrofungi. They are commonly used in various ways and form one of the choicest table dishes. They are liked for their flavor, besides being delicious. Mushrooms generally grow in the dense forests, meadows, and open pasture lands, especially during rainy season. Mushrooms hold a fascination because of different forms, shapes, and colors. Most of the mushrooms are edible, whereas few species like Amanita are poisonous. Globally, scores of mushrooms are being cultivated, but in India, only 3 types of mushrooms are cultivated. They are white button mushroom, oyster mushroom, and Chinese mushroom, which is also known as paddy straw mushroom. For artificial cultivation of white button mushroom, the basic requirements are spawn, also known as mushroom seed, compost, casing, crop management. Spawn is the vegetative growth of mushroom on a suitable medium and is equivalent to seed in other crop plants. It can also be prepared in polypropylene bags. Spawn production basically involves 3 steps: raising of pure culture, preparation of master culture, mass multiplication. For raising pure culture from spore, selected mushroom food body is mounted on a wire and placed in sterilized Petri plate covered with a glass jar. The spores thus collected after 24 to 48 hours are transferred to the culture medium under aseptic conditions. The inoculated tubes are kept at 22 to 26 degrees centigrade in an incubator for 12 to 15 days. Tissue culture is another way of raising pure culture. The selected fruit body is surface sterilized with a cotton swab moistened in 50% ethanol. A small bit of trammel tissue is transferred to culture medium under aseptic conditions and incubated at 22 to 26 degrees centigrade. The pure culture is ready within 12 to 15 days of incubation. For preparing master culture, a bit of agar block along with mycelial growth from pure culture tube is transferred to sterilized bottles containing suitable substratum and incubated for 15 to 12 days. For mass production of spawn, 0.5% calcium carbonate and 1.5% calcium sulfate are mixed thoroughly with partially boiled and dried wheat grains. Then it is filled in glass bottles or polypropylene bags. These are then plugged with non-absorbent cotton and sterilized at 22 pounds pressure per square inch for 90 minutes. After cooling, these bags are inoculated with 20 to 30 mycelial coated grains in each bag from master culture under aseptic conditions and incubated at 23 to 25 degrees centigrade. The spawn bags become ready within 12 to 15 days. The qualities of good spawn: strandy growth of the mycelium, free from contamination, no slimy growth, and proper mycelial coating on grains. For growing button mushroom, compost is needed. Compost is a selectively prepared medium which promotes mycelial growth of mushroom to practical exclusion of undesirable organisms. Compost can be prepared by long method or short method. The ingredients used in long method are wheat straw, can, urea, superphosphate, gypsum, wheat bran, molasses, furadun, BHC dust. The wheat straw is thoroughly wetted with water. A mixture of wheat bran and fertilizers is wetted, kept covered with moist gunny bags for 24 hours. This mixture is evenly distributed over the wet straw. A pile measuring approximately 4 feet wide, 4 feet high, and of variable length is made with the help of 3 wooden or iron boards. Pile is turned periodically after 48 to 72 hours intervals to prevent anaerobic composting and compensate for water loss through evaporation. Compost becomes ready within 27 to 30 days. The ingredients used in short method are wheat straw, poultry manure, wheat bran, urea, gypsum. The wheat straw is thoroughly wetted so that every bit of straw becomes wet and absorbs sufficient water. The wet straw is put in a layer over which poultry manure and wheat bran are evenly distributed in layers. Another layer of wet wheat straw is laid and trampled hard by pressing with feet to encourage anaerobic conditions. A square-shaped stack is prepared by adding 3 to 4 layers of the ingredients. The stack is given 2 turnings at 48 hours intervals. The whole mass is broken and water added if needed. A standard pile is made with the help of wooden board and urea is added evenly on the 5th day. In all, 4 turnings are given in phase 1 and gypsum is added in the 2nd turning. On the 10th day, compost is filled in the bulk chamber for pasteurization and conditioning, which is also known as phase 2 or indoor composting. The bulk chamber is fully insulated. The aerated steam or fresh air is passed from the grated floor of the tunnel. Compost is pasteurized at 59 to 60 degrees centigrade for 2 to 3 hours and conditioned at 50 to 55 degrees centigrade till ammonia smell is gone. The ready compost, which is dark brown in color, is tested in the laboratory. Qualities of compost. Compost should be dark brown in color, free from ammonia smell, moisture 68 to 70%, pH 7.0 to 7.5, nitrogen 2.2 to 2.5%, not greasy or sticky, free from fungal competitors, insect pests and nematodes. The spawn is thoroughly mixed in the ready compost at the rate of 0.6% and filled in polythene bags. The spawn bags, covered with newspaper sheets, are kept at 22 to 26 degrees centigrade and 85 to 90% relative humidity. The white mycelial growth impregnates the compost completely within 12 to 15 days and becomes ready for casing. The process of covering the spawn-run compost with casing material is known as casing. The casing material consists of well-rotten farmyard manure and loam soil mixed in equal volume. This mixture is pasteurized in a chamber at 65 degrees centigrade for 3 to 4 hours. Casing is done to avoid moisture loss and to initiate fruit body formation. A 5 centimeter thick layer of cool, moist casing material is applied on the surface of the spawn-run compost. Similar environmental conditions as during spawn-run period are maintained. After 8 to 10 days of casing, white mycelial growth is seen on the casing layer. It is time for introducing fresh air. Fresh air is introduced with the help of perforated polythene duct to provide 3 to 4 air changes per hour and bed temperature lowered to 16 degrees centigrade. The pinheads appear after 5 to 6 days of introducing fresh air and develop into buttons which are ready for harvest in next 3 to 4 days. The relative humidity of 85 to 90% is maintained in the cropping room by daily water spraying. The mushrooms are harvested by slightly pressing and twisting with the help of thumb and fingers. The lower end with soil is cut off. It is needless to say that mushrooms are nutritious and contain proteins 3.8%, fats 0.3%, carbohydrates 3.5%, fiber 1.0%, ash 1.2%, water 90.2%.
Amongst SAR countries, Dr. Y.S. Parmar, University of Horticulture and Forestry at Solan in Himachal Pradesh, has taken the lead in scientific research, technology generation, refinement, and dissemination of mushroom cultivation. Because of the search and extension efforts of our experts, mushroom growing is not only becoming popular throughout India, but many neighboring countries are also taking the advantage of our experiences. This natural product has both consumptive and productive use values. Mushroom provides rich, nutritious, and delicious food to all sections of the society and are also in great demand both in domestic and foreign markets. Thus, it is an ideal, eco-friendly, income-generating activity for the farmers.
The Indian Council of Agricultural Research established a national center for mushroom research and training at Solan itself. The mandates given to this national facility were to conduct basic and strategic research and applied research also on different types of mushroom for developing their technology of production and also utilization of the wild germplasm of mushrooms.
Oyster is the other